Good evening. Good evening. My name is Daniel Colon Ramos, and on behalf of my fellow organizers, Rudinger, Kathy, and Samantha, I would like to welcome Catherine Dulac to as our keynote speaker this evening, and just a few words about Dr. Dulac. So Dr. Dulac's work focuses on understanding brain mechanisms underlying the control of social and sickness behaviors in mammals. Just a little bit about her background. She grew up in Montpellier, France, and graduated from the École Normale Supérieure in Paris, where she received her PhD at the University of Paris. And she was a postdoctoral fellow with Richard Axel in Columbia University, where she started looking at pheromone receptors and their sexual dimorphism, work that she later pursued upon joining the faculty at Harvard University in 1996. She was chair of the Harvard Department of Molecular and Cellular Biology from 2007 to 2013, and is currently the Samuel W. Morris University professor in that department. And I don't think her work needs much introduction, and we're about to see it, but I do want to say a few words about it. And I'd like to mention that as a person that has been, enjoying her lectures and her work for many years now, one characteristic is that it beautifully combines cutting-edge molecular biology approaches to tackle what are considered systems-level questions, many times pioneering methods that didn't exist before in genetics, transcriptomics, physiology, and imaging approaches. And in doing so, her group has uncovered neuronal bases underlying instinctive social behaviors. They have also uncovered a set of brain functions that are, and this set of brain functions are typically impaired in mental illness. Her research has helped decipher the unique characteristics of social recognition, including sensory cues that trigger distinct social behaviors, including nature and identity of social behavior circuits in males and females. And in recent work, which I think she'll speak about tonight a little bit, her group has discovered how brain-specific circuits direct adaptive changes in behavior during sickness episodes and encode social isolation versus social groupings. I mean, really complex systems-level questions that they have been able to beautifully dissect at the level of circuits and molecules to understand this regulation. Her work has been recognized, and she has been recognized with numerous awards. She's a member of the U.S. National Academy of Sciences, the French Academy of Sciences, American Physiological Society, and she recently became an officer of the Légion d'honneur in France. So without further ado, welcome. We're very excited about your lecture.
Thanks a lot, Daniel, for the very kind introduction. Thanks for the organizer for inviting me to give this keynote lecture. It's really a special treat, actually, for me, because I actually did my PhD in developmental biology, and Cold Spring Harbor was the first place where I attended an international meeting. In fact, it was the first time I traveled to the US from France. And, it's interesting. I didn't learn English in school, but when I knew I was going to go to the US, I bought a book and some tapes, and for 3 months, every day, I learned one lesson, went through one page, et cetera, et cetera. So when I landed here, I really remember stepping my foot on the US soil and thinking, holy shit. I had never, ever spoken to anybody, because when you have a tape, you talk to yourself, right? So you can imagine that my English was probably not too good. I don't know how it is now. I assume it has improved a little bit. In any case, I was a developmental biologist. The meeting, I was checking with Larry Zipursky, the meeting was probably one of the first meetings in developmental biology, not that serious here, maybe quantitative biology with a focus on developmental biology. But it's really interesting because as I was attending this meeting, I could sense the same vibe, the same enthusiasm of the trainees. And so there's really something very special about this Cold Spring Harbor meeting. It's also interesting because as decades have passed, some of the essential questions, some of the essential concepts are still there, still, topic of inquiry. But the experimental approaches and some of the intellectual thinking really has changed tremendously. So anyway, if somebody would have told me at that time in my first meeting at Cold Spring Harbor, I would be here, give a keynote lecture and be a professor in a university in the US, I certainly would have told them they were full of it. Anyway, so, I had a longer title on but I realized I had assumed the keynote was an hour, so I had assumed that I would talk about circuit much more. But I will focus, for today's audience, I will focus on the development of behavior circuit relate to instinctive behavior, social and sickness behavior. And so my lab, as Daniel kindly summarized, my lab has been interested in trying to understand the circuit underlying instinctive social behavior, and we've been using molecular, cellular, circuit behavior approach, any type of approaches, to enable us to uncover the specific neuronal population and the associated circuitry that trigger a number of instinctive behavior, social behavior, and more recently, sickness behavior. And so over the last few years, we, for example, identify specific neuronal populations that are essential for parenting behavior, both in males and in females, and the associated circuitry. We identify a set of neurons also that trigger a large array of behavior changes that are associated with inflammation or sickness. And more recently, we also discover specific cell type and associated circuitry that encode the state of the animal that is either in social isolation or together with other members of the group. And so the question we are now interested in addressing is where are these neurons coming from during development? What is the process of development? And in particular, because we are dealing with this instinctive behavior, as many of you know, there has been decades of debate about the respective role of the environment or genetic programming of those behaviors. So this is the type of question we are interested in. Now, in the background of this is all the new approaches of identifying cell types. And it's quite interesting that last year, the entire mouse brain transcriptome was described in an issue of Nature with 5,000 transcriptomic cell types that were identified either both using single-cell sequencing or transcriptomic approaches. But interestingly, if you look at where most of the cell types are, relatively few are associated with the cortex, and the majority are in subcortical areas. And in fact, around 900 cell types are located, were found in the hypothalamus, which is the region where we found the cell types that control the various instinctive social behaviors. And so one idea is that in the cortex, there are relatively fewer cell types because they are related to general purpose circuitry and enable flexible computation. And by contrast, in the hypothalamus, there are a very large variety of cell types that are more dedicated to the control of specific behaviors. But certainly, that makes our problem even more complex. How are these cell types emerging? How are they defined? How do they know? How do they acquire their fate in controlling specific behavior? So the particular area of the hypothalamus we're interested in is a hotbed of behavior control. It's called the preoptic area. And it's an area that is both functionally and structurally extraordinarily diverse. It controls an array of social behavior, for example, mating, parenting, social interaction in general, aggression, but also a set of homeostatic function, thirst, thermoregulation, sleep, feeding, and so on. And these various functions are spread throughout a number of sub-regions, such that no sub-region is specialized in any particular function. But to the contrary, any given sub-region seems to be involved in multiple of these homeostatic and social function. And so one idea is that the neurons that are involved in the control of this behavior actually talk to each other in order for the animal to set priorities in function of their state. So if you sleep, you cannot be parenting. If you're parenting, you're less sensitive to hunger, etc., etc. So the idea is that all these circuitry are really intertwined very closely. So a number of years ago, together with my colleague Jiawei Zhang at Harvard, we decided to establish an atlas of cell type in the preoptic area and identify not only the identity of cell types, but also their location in the preoptic area using this in situ transcriptomic approach called MERFISH. And these led us to identify 70 neural population, mostly undescribed before. And by combining the immediate early gene CFOs after behavior, such as aggression, parenting, and mating, we were able to assign one or multiple or zero function to some of these cell types. And so these, together with work from other authors, we have quite a range of cell types that have been identified in this area that are involved either in the control of various social behavior or various homeostatic function. So how are these cell types emerging, and how do they come to adopt certain function in young animal? So P0 is birth, P18 is weaning age, P40 puberty, and then adulthood. What is quite interesting is that all the functions I described are actually very different in young animal rather than in the adult. So before weaning, the young animal is really depending on mom for everything. For social behavior, they huddle, they suckle. For homeostasis, they need food, they need thermoregulation through mom, and then they do not explore. Later, after weaning, they start to investigate, they start to eat, drink independently, they start to have their own thermoregulation, and they start to explore. And then only after puberty do they adopt adult sex-specific behaviors. So the question is, as these functions change so dramatically in postnatal to adult stages, not only do we want to know how these cells come on board, but how are these changes reflected in the trajectory of these cell types? So what Harris Kaplan, a terrific postdoc in the lab, did, and the work I will describe in this talk is primarily from his work, he decided to perform RNA-seq sequencing from various stages, representing late embryonic stages, E16, E18, early postnatal, P0, P4, P10, weaning stage, P28, and then adult, P40 and P65. And so what I would like to describe is the early life, the emergence of homeostatic independence, the emergence of sex differences, and then the role of environmental input in this developmental program. So so Harris dissected and freeze brains, 3 brains per age and per sex. We did males and females. We dissected the hypothalamus in frozen tissue using anatomical landmark, and then performed single-cell sequencing paired with ataxic in duplicates from each stage and each sex, leading to around 300,000 nuclei, both RNA-seq and ataxic, across over 35 samples. So what we obtain by analysis of these data is around 150 cell types in the adult, with 83 GABAergic neurons, 64 excitatory neurons. The cluster are shown here, with excitatory, inhibitory, and various non-glial cells. What is interesting is that this clustering here encompass all the different stages in which the cells are represented by a color code. Very light represent early stage. For example, this yellow pink here is 16, and then as we go darker to the blue, we go to older ages and adult. And what is interesting to see is how the various ages are intermingled with each other, and such that if we look, for example, at the close-up of this cluster here, we can see that each cluster has this interesting temporal gradient of cells that are ordered according to age. So from early and then embryonic, early postnatal, young adult, and full mature adult. And so to recognize the identity of these cells compared to previous data, we perform a label transfer of P65 cells into published atlases. And these enable us to identify not only cell type according to previous description, but also a cell type that had been functionally identified. And so what we found is that we can recognize cells involved in parenting, mating, social behavior, thirst, sleep, etc. And so the question is, how are these different cell types emerging in young animal? And interestingly, if we look at this cluster here and use the information from the label transfer, we can recognize that the various adult cell type can readily be recognized in this mixture of cells from adult and early stages. So, for example, this cluster corresponds to neurons known to regulate thermoregulation. This one, neurons involved in parenting, and this one in dark blue, neurons known to control thirst. So this is quite surprising, which is already at stages as early as E16, we can assign the cells to a particular functional cluster. So how does that come about? Well, if we look and perform this comparison matrix between the cell types at E16 to the adult at P65, we can recognize very clearly the full complement one-to-one of the 150 cell types that were identified at P65. So all the cells seem to be recognizable. Moreover, if we look now at the subregion of the preoptic area that are here labeled with a color code, we can see that at P65, neurons that belong to a particular subregion here with the corresponding color code seem to be very similar to each other. So this is P65, and interestingly at E16 you can see that already the cells, the similarity of the cells seems to coalesce around this subregion. And so it seems that at E16, both the cell types that of the future adult cell types, but also the subnuclei can already be recognized. So that was surprising to us, that already as early functions that are used way, way later in the animal, in the adult, such as mating, parenting, etc., are already identifiable in the embryo. And similarly for homeostatic functions such as thermoregulation, thirst, eating, etc., that are also used much later. So this prompted us to look at an even earlier stages, E14, in which we thought that maybe we could see some type of differentiation process. And so when we look at cells at E14, we can see, based on markers of cell cycle as well as markers of progenitors and postmitotic neurons, that these cells can be subdivided into two groups, neurons and progenitors. And when we look at the neurons themselves and do again this correlation matrix between the adult cell types and E14, you can see that here again, the cells can easily be identified. Not all of them, though. It's very clear that if we look at the corresponding clusters, some cell type, for example, the one located here, correspond to a cluster associated with a substructure called the AVP-MNPO, which is important for thermoregulation and thirst. This is already very clearly differentiated at E14. But these clusters here, this set of clusters that correspond to the medial preoptic nucleus, which is an area very involved in specific social behaviors, this is not yet completely differentiated and is poorly recognizable. At E16, however, as you can see, these clusters are perfectly identified. So it seems like within 1 or 2 days, from E14 to E16, the cells are able to rapidly diversify. So what if you look at the progenitors now? So if we look at this other part of the clusters here, then we find about 26 subpopulation. But when we look at what these cells are, we can find possibly 2 groups. One group here that express markers of progenitor cells, and another one that express DCX here, perhaps identifying migrating neurons. And these cells not only do not have any cell identity, but they also have absolutely no identity of cell region. So in summary, we go from E14 to E16 to already have the entire repertoire of adult cell types. And so what's going on afterwards? And so what Harris decided to do is to visualize using this approach, the distance to adulthood. And so you can see here a color code of the various ages. And what he did is to calculate the centroid in high dimensional space of the cell type at any given age to the centroid of the adult cells. And so this shows that the cells are very distant from the adult. P4 is less distant, P18 is closer, etc. And so these provide us an idea of the trajectory of the cells. And so if we do this for all the cell type, here shown only on excitatory neurons, you can see that each cell has its very specific trajectory, and that trajectories are really quite variable from one cell type to the other. And there are different things, different characters that differ between these cell types. One is the timing to maturation. There are cells that start their maturation at P0, and some cells that just start to mature very late at P28. There are trajectories that are very abrupt, and there are trajectories that are slower. So this is the distance to adulthood in function of time. This is the average that you see in black, and you can see the cells here below are cells that are very distant from the adult at P0, and then have this very slow trajectory that takes quite a while, whereas these cells in purple here start at P0 by being very close, already quite close to adulthood, and has this shortened trajectory. And so when we look at the determining times, when are the big jumps in these distances, we find that P10 and P18 right here, as well as P28, P65, so weaning and puberty, are really the 2 stages in which there are this big jump in maturation. So in other words, these very major changes in the distance of a given stage to adulthood. And so what are the determinants of these different trajectories? There are 3 that we could identify. One of them is the location. So if we look at these trajectories according to the subnuclei within the preoptic area, we found that nuclei such that the MPN, VMPO, or the paraventricular nucleus, as you can see, are very distant from adulthood at birth and have this very slow trajectory. And other areas such as VLPO and AVP-MNPO are closer at P0 from adulthood and have this faster trajectory. So I assume very few people in the audience know what these various subregions are, but I can tell you that MNPN, PVN, these are areas that are mainly involved with the control of social behavior, and VLPO, AVPE are areas involved in homeostatic functions such as sleep, or thirst. And so these intrigued us and led us to look now specifically by function for the particular cell type for which we were able to identify function. And what we found is that the cell types that we sorted out of this data that are specifically known to be involved in the control of social behavior have this very slow trajectory and start very distant from adulthood at P0 and then slowly over time acquire their adult function. So these are all of them. This is just a subset of them that are involved in parenting, aggression, social recognition, and then neurons expressing oxytocin and vasopressin. When we looked at neurons involved in the control of homeostatic function, we found a bit of a mix. There are some that have very early differentiation and some that have late differentiation. And overall, I gave an example here of some with very quick differentiation that are involved in third, sleep, and then neurons from the suprachiasmatic nucleus that are involved in the circadian clock. So again, some interesting difference of cell trajectory according to function. Now, you might say, well, okay, what does that say? I found this data really intriguing because a cell at P0 or P4 or P10 does not know it will become a neurons involved in the control of parenting behavior or male sexual behavior or aggression. So somehow there's either some genetic programming to that cell, some type of lineage information that tell them, hey, you need to go slow and that will be your developmental trajectory, or there are specific input or specific type of signal that targets those cell types specifically and modulate their timing of differentiation. So the third really important parameter that determine the trajectory is sex. And so these are the trajectories that are divided, this time for inhibitory neurons, divided for cell types identifying males and cell types identifying females. And this is just to show you how different the shapes is. You can see here you have this very early development occurring already at P0 or P4 that emerge in male, but very few of those are apparent in female. These are some key examples of the same cell type in females and in males. You can see again in males this development that starts very early, at very early stages, and start way later in females. So first of all the cell types are found both in males and females, and this early start of development of this subset of cells that are found in male and not in female is likely to be due to this early testosterone surge that occurs in male and not in females. And in fact, if we look again, the distance to adulthood and the changes in these distances, you can see that in male, there is a significant change occurring between P4 and P10 that does not occur in female. And this, again, is likely to be due to this perinatal testosterone release. And so this is an example of cell type that is not sexually dimorphic. This is an example of cell type that has very different trajectory between males and females. So just as a reminder, in males, you have this perinatal surge in testosterone that then goes down and then resume again around puberty, while in female, the surge in estrogen occurs only at puberty. But importantly, testosterone acts in in mice, mainly through the isomerization by aromatase into estrogen, and it's the estrogen that is the active element that masculinizes the brain. And estrogen can act directly on a cell that has aromatase or act in a paracrine manner on neighboring cells that express the estrogen receptor. And so when we look at the expression of estrogen receptor, androgen receptor, and progesterone, etc., we found that estrogen in these different cell types, as you can see, is for some of them, a subset of them, expressed extremely early, as early at E16. So these cells expressing the estrogen receptor are primed to be sensitive to local estrogen. And then many of them have expression later on. The coding here in purple indicate that these cells are all involved in the control of social behavior. These are cells expressing androgen receptor. As you can see, these appear relatively later, and it's very low perinatally and then substantially increased with age. Interestingly, if we look at aromatase expression, we found quite a diversity of expression. Some cells express extremely early and sort of consistently throughout age, some very transiently, some very late. And interestingly, some do express estrogen receptor and some do not express estrogen receptor, suggesting that these very interesting paracrine mechanisms that I just mentioned before. So what are the most striking sex differences? Well, we found about 15 cell types that have a lot of gene expression differences. This here is calculated on PCA distance between male and female transcriptome. This is the number of sexually differently expressed genes. These cell types have mainly all been described by in the literature by Jessica Tolkien here or Nira Shah and other groups. We found that all these cells express a lot of estrogen and various steroid receptors. And if we look at when they start to be different in this PCA analysis between males and female, we found that 4 cell types start to show sex differences already at E16 to P0, which was quite surprising to us. And then these differences expand. There are way more cell types that emerge and further develop after puberty. And these, for each of these cell types, again in purple are cells involved in social behavior. You can see the number of sexually differently expressed gene, sex, DEG. And as you can see, most of the sex DEG are found at P4, P10, and then the differences seem to be lower. And what is interesting, if you look at 2 cell types that are extremely sexually dimorphic in terms of gene expression, and you look at what type of genes are different, the program in each cell type is very unique at any given time. So, for example, for this cell type P4, P16, you find genes involved in dendrite, neural projection, cell junction, and then a little bit later involved in synapse assembly, serotonin receptor, etc., and then even later other type of function. And this other cell type has a very different program that we can look at and try to understand how these cells acquire sexual maturity. So finally, the last question I would like to address is, all these cell types control instinctive behavior and homeostatic function. So how does life experience affect their development? And this is a very interesting question because on the one hand, we all know all the big debate between developmental psychologists and from the US and ethologists from Europe that fought over what is instinctive and not. None of these issues has ever been solved. But one thing is sure, is that in terms of behavior, social interaction are absolutely essential. And, this very famous experience from Harlow in the 50s, where he isolated young monkeys, showed that this animal never developed normally. This is also a time where people were observing women who were in jail, whether they were able to keep their baby or not, and observe the difference in behavior afterwards. It's very clear that early social interaction is absolutely essential. And then even more recently, these extensive studies that have been done in Romanian orphanages, in which infants were literally not really abandoned because they were taken care of in terms of food and drink, but there was nobody to interact with them socially, really had extreme impairment in behavior, in social behavior. So on the one hand, it seems pretty clear that there might be, in the same way as what was found in the visual system, some type of critical period for social behavior. But on the other hand, nobody has ever looked at it. People have thought a lot, but nobody has looked at it. And so I'm mentioning here the recent work from Larry Zipursky that looked at cell types in the visual cortex in normal mice and in dark-reared animals that show a defect in cell type development. And so similarly, we can ask in mice, if we look at animals with various social impairment, various sensory impairment, do we have a defect in the development of this cell type involved in instinctive behavior? And so luckily we really have a lot of choice to look at sensory impairment. We looked at impairment in somatosensation by using 3 types of mutant, the CDX, the piezo2 restricted to the higher brain. These animals do not sense touch. These adrenaline Cree, GABA or B3 have the opposite. They have touch hypersensitivity. The TRPM8 mutant are impaired in cold sensing. We also use olfactory mutant that are strongly impaired in social behavior. TRPC2 mutant that are impaired in vomeronasal function also have strong defect in social behavior, particularly the sex specificity of social behavior. And then, to some extent as a control, animals that are dark-reared, that to my knowledge have no defect in social behavior, but might have a defect maybe in homeostatic function of some sort. And so we perform single-cell sequencing at 4 different times, P10, P18, puberty, and P65, on those various mutant and sensory deprivation conditions. And when we look at the transcriptome of this cell type and compare mutant versus wild type, we were quite surprised. So as you can see here, these in color indicate the cell types that have significantly different transcriptome in mutant versus control. As you can see, there's really very little, if no, changes in somatosensory mutant, except perhaps the cold sensing mutant that have a cell type that might be different. But we have a major set of differences in TRPC2 mutant, so in mutant for pheromone sensing. Now, the number of cell type that is affected is actually so high that we were concerned that there was something wrong there. And so we actually sequenced the visual cortex of this TRPC2 mutant to see whether there was something specific to the POA or not. And we see absolutely no difference in the visual cortex. So it seems like vomeronasal function is absolutely essential for the normal trajectory of these cells. What is different? Well, what we found, first of all, if we look at the various cell types that are affected, here again in green are cells associated with homeostatic function, and in purple social function. As you can see here, the mutant has defect that mainly, but not only, affect cells associated with social behavior. So these are cells involved in homeostasis. As you can see, a number of them are affected compared to control. We also found that if we look at the difference in the transcriptome at different ages between the wild-type control and the TRPC2 mutant, the major differences are between P10 and P18. And interestingly, it seems like the transcriptome becomes almost normal at the adult stage. So it seems like there is a delay in preoptic area maturation. I won't show you those data. You know, it's possible that the developmental trajectory could go completely wrong or simply be delayed. And we were able to show that there is, in fact, a delay of several days. And when we look at adult cell type, we found that a number of cell type in the adult in the TRPC2 mutant are also affected, suggesting that two things. One is that pheromonal input are essential for the normal trajectory of neurons from the preoptic area. And second, that this time window, P10 to P18 right there, might potentially represent a putative critical period for preoptic area development. So what we found is after identifying all these cell types in the adult and asking how these cells coming about molecularly and hopefully in the future in terms of function and neuronal dynamic, we found this really interesting program with early diversification, aerolization, so the formation of this subgroup, and then some activity-dependent influences in a particular time window, and then a functional maturation, so the cells acquiring its final characteristics that are very cell-specific according to the future function of the cells. And so this is quite interesting because, as Timbergen said, in order to understand the evolution of behavior, you have to look at development. And what we found is that there are widespread external influences, both hormonal through steroids, sensory influences, as I mentioned, from the Vomeronasal system, but also some type of lineage inherited information, in particular cells that will fulfill future function according to those functions have very different trajectory. And on this, I would like to thank the people who've done the work. The major part of the work has been done by Harris Kaplan, a postdoc in the lab who is now on the job market. He's spectacular. I really encourage you to check him out. Brandon Lodgeman, another postdoc, Tate Yavitz, who was a master's student for 6 months in the lab and now is a graduate student at Columbia, and then our collaborators and source of funding. Thank you.
That was a wonderful talk. Thank you very much. I was wondering, and sorry if you already covered this, thinking back over now several years of comparing cell types in the motor cortex, one potential top line is that through the chosen mathematical framework, there's less variation between species than we might intuitively expect. So given that, was there more or less transcriptomic variation across the lifetime of the animal than you intuitively expected going into the project?
I don't really have an answer to that. I think, however, that cross-species comparison is going to be extremely important and extremely interesting. But I don't think there's been much of this. Gilles Laurent has performed some transcriptomic studies across variety from reptile to mice to primate, and found differences both in the cortex and the hypothalamus, and really generating the idea that it's not that the hypothalamus is an ancestral area and the cortex is a new area. Actually, everything evolves together, which I think is super interesting.
Yeah.
Yep.
Over here. So I was really fascinated by the differences in developmental timing, depending on the cell type, and that seems to be also somehow related to some kind of spatial patterning, let's say. On the other hand, you said rightfully that it could have cell intrinsic origin or depend on the input to the cells that maybe indeed have a spatial pattern. But then on the other hand, at the end, you show that changing a lot of the sensory input does not seem to influence the developmental timing, with the exception of vomeronasal organs. So do you think it's the VNO, actually, that may change some, but not all, of the cell type-specific developmental timing? Or otherwise, can you think of other extrinsic cues? Although, of course, that would be my personal bias. It could also be the GRN of these cells and an
Exactly.
intrinsic difference that maybe is what seems to emerge from your data.
This is a super interesting question, and there are several hypotheses, and I really don't know. So first of all, quite a number of the cell types that are affected are involved in social behavior, and we expect them to receive input from the vomeronasal system. So it's possible that these cells no longer receive active input and therefore don't develop normally. And if that's the case, then some of the phenotype of the TRPC2 mutant might in part due to the defect in this development. But there are a lot of cells that have nothing to do with vomeronasal input, that are involved in homeostatic function, that are also affected. And so these, I think, might suggest that there might be some hormonal or paracrine phenomenon happening. It's possible that the neurons that receive vomeronasal input in turn release goodies for their neighbors, and if the goodies are not there because those cells are not active, then overall the entire development of the cell type, irrespective of whether they receive vomeronasal input or not, is impaired. Alternatively, there might be just one or two key nodes that are simply impaired. So, for example, the GnRH neurons come from the olfactory placode and migrate along the vomeronasal nerve. I actually don't know whether GnRH neurons do develop in TRPC2 mutant. There are so few of them that we don't see them in our data set. So I don't know whether they are there. And if they are there, I don't know how abnormal they are. These cells, and maybe other cell type of that sort, you need maybe 1, 2, or 3 that release hormones or neuropeptides that might be essential for the development of their neighbors. That's the type of thoughts we have. But frankly, these are just really hand-waving.
Thank you.
But it is very surprising.
Hello. Your work has a really interesting ability to kind of connect psychology with biology. And so I'm wondering, in some ways, since there's this impact on hormones and this idea of this potential paracrine signaling, if you've looked at cortisol and particularly stress, and how, since that's related to progesterone and then therefore estrogen, if there's any difference in this kind of trajectory.
Yeah, so we haven't done any of these because, this is brand new. But I think that the question of how the environment and, for example, stress, maybe sickness, or maybe, all sort of environment would affect the development of these cell types is super interesting. As you say, through cortisol, could affect progesterone, could affect all sort of signaling that affect the trajectories of these cells.
Hi. Awesome talk. I really liked how you were able to essentially map the embryo cell types onto adult cell types. One aspect of your introduction that I found really interesting is how you talked about how the behaviors kind of develop as well, and you have these young animal-specific behaviors and then the adult animal-specific behaviors. I was curious if you thought that the young animal-specific behaviors were maybe mediated by an unknown cell type, or if that was maybe the ability of, or if an adult cell type could maybe do those young behaviors and then develop that adult behavior later in life, and if that may contribute to its slower diversification.
Yeah, so this is actually what Harris wants to do in his new lab once he gets a position. But I think one little caveat in the description I gave to cells involved in social behavior that develop late is that all the social behaviors that are now studied, described, are all adult social behavior. And there has been very, very little, except recently by 1 or 2 groups, some attempt to look at newborn or postnatal behavior. And I completely agree with you. This is just a really interesting question. What are the cell types? How do they change? Are they the same cell type but function differently, maybe different wiring? Are they different cell type that then either disappear or do other things? Yeah, this is super interesting.
Hi. Over here. Great talk. This might be a little bit of a naive question, but just quick question on like the transcriptomic analysis. So when you look at like the rate of differentiation across different cell types, I was just curious how you normalize for like the onset of neurogenesis. Like I assume like not all these cell types are being born at the exact same time. So how can you distinguish whether like a cell type has like a prolonged rate of differentiation versus it's just being born later and it's just like at a earlier stage in differentiation?
I'm not sure I completely understand the question. You know, we have these clusters, right? We have this data in really high dimension, and we, based on specific markers, we can attempt to identify, to give a name to one of these clusters. If it has a lot of genes associated with progenitors or cell division, you know, we can define a cluster as being dividing progenitors. If it has a marker of postmitotic neurons, that's what we have. And because we look at the same cell type over time, and, you know, we're not looking day after day. We're looking at several days apart. So the clusters are different enough that we can have a sense of the trajectory.
Thank you.
Talk. I have two questions. First, when you are characterizing the sexual dimorphism, I noticed that it seems to be a cluster of cells that called EP2 that is called only different at the early stage between the two sexes, but they tend to be the same later stage. So could you elaborate on what that would be?
We don't know. Yeah, it's very intriguing. This group of cells that seem to be different between the sexes very early. First of all, this is quite surprising. Somebody, an author, has described this in the literature, but had never been described using single-cell sequencing. So we need to dig into this a little bit more.
My second question is that when you are characterizing the impact of earlier perturbation, for example, the somatosensory defects, and how that impacted the hypothalamus neuron later on in the development, you did like cell type characterization by RNA-seq. I wonder if you can also evaluate a functional output with a function, because I wonder, even though you don't see a cell type difference, could that affect the connectivity or like in other way impact them other than cell types?
Yeah, we haven't done that, but I agree. And in fact, based on some transcriptional differences, we can assume that, for example, the number of the synaptogenesis is different, the connectivity is different in general, as well as the excitability of the cells, etc. I mean, you know, this is an enormous data set, and hopefully it opens a lot of interesting questions like the one you mentioned.
Thank you.
Thank you, Catherine, for this beautiful work. I appreciate that you're going back to your developmental origins and you know, maybe following Cajal's advice to study the young wood.
Yep.
So my question is about the molecular relationship between these different populations, right? So what if you were to go back in time to, let's say, E13? Would you find maybe like a mother progenitor of all social neurons or homeostatic neurons?
You know, that was one of the dreams I had. And, you know, we went up to E14 and couldn't find that. So, yeah, we can go earlier. It's complicated, right? Because the cells are also migrating. The brain area that we need to dissect is very poorly defined. And, you know, it's yet another huge experiment of sequencing to do. So it's a very good idea. I'll leave it at that. Hopefully somebody will do it.
Okay, well, please join me in thanking Catherine Dulac for